everything is being live recorded here right now. Okay, so please be very careful with your behavior in this class. Why are you so sitting on those back benches? Come in here. Come in front. Come in front. Last row also. Come in front. Don't leave any space. You also, girls, come in front. Come somewhere in front. Be careful about whatever you are doing. Everything is being videographed today, and there are inspectors going on here and there. You must have noticed by now that a lot of movement is there today. Okay. Okay. I will try to cover up the time which is lost. Okay. See, we are going to discuss the topic of immunity today. It is in two parts. Today is one lecture, and next Monday there will be another lecture, same time. So, what do you understand by immunity? Can you hear me without the mic? Can you hear me without the mic? Yes or no?
No, sir.
Oh. What is the meaning of the word immune? Immune. The word is immune, and immunity is a form of activity which is arising out of immune. So it is called as immunity. Immune means to cover, or immune means to envelop something. Like you are covering a book with plastic cover, so now that plastic will immune the book against water. You can say it is a proofing, waterproofing, something like that. That is the actual meaning of the word immune. Okay. So in our body, there is an immune system which will protect us on a daily basis from foreign bodies, which includes living as well as non-living. Non-living means chemicals or toxic substances which are there in our environment. Commonest toxic substances are pollution products, automobile pollution or other industrial pollutions. Okay? And living are bacteria, virus, fungi, all these things. Is that clear? Because of that we require immunity. Most of the immunity is for this. In addition, there is another condition in which the body will behave with its own cells as if it is a foreign body. That is another concept of immunity, and that is called as autoimmunity. Auto means self. Okay? Understood what is the basic meaning of immunity? Now, you know there are WBCs and RBCs, and in WBCs there are different types of cells. In different types of cells there is a agranulocyte type of cell. Of that, one is lymphocyte. Okay? So all the immune mechanisms are mediated by lymphocytes. Okay? Till this system starts, and I can show you the slides, you can write on one paper from the top to bottom. It will be a flow chart type of information of this entire lecture, so that you can later on revise it through that way. So first line you write immunity. Just below the immunity you leave two lines, and in between those lines you write living organisms and non-living organisms, non-living items. In living organisms you write bacteria, virus, fungi, and non-living you write pollutants, industrial products and toxins. Okay. Now immunity is principally governed by a type of WBC which is a granulocyte, and in that the specific type is T lymphocyte. Okay. Now below that first arrow you write T lymphocyte. Then draw two arrows, right and left. On the left side you write cell mediated immunity. Okay? Cell mediated immunity, also called as CMI. It is done with the help of T cells. Type of lymphocyte. Two types of lymphocytes are there, T cell and B cell. So this is by T type of lymphocyte. On the right side, ठीक है, Is it visible to all? Is it visible?
Yes.
Now see, you can read this, don't try to copy, I will be telling you specific points which you can note down. So in cell mediated immunity, the principal cell is T cell. Okay? It is a type of granulocyte which is a type of WBC, white blood cell. Now they will give immunity against all living organisms like bacteria, virus and things like that. Okay? What exactly happens when a foreign body enters our system? All the cells are having a cell membrane. All cells, either bacteria or virus. On the cell membranes are present specific proteins. These proteins are called as antigens. When they enter our system, they are taken up as a foreign body because that antigen is normally not recognized by our system. Okay? So our body will take that antigen as a foreign body or as a invading organism. So the system here, this cell will try to protect our body from those antigens by a specific mechanism. That is called as cell mediated immunity. Okay? Can you follow this? Can you understand this? Foreign body, cell membrane, antigen identified by our system, and then the following activities will take place. What are those activities? They are the immune related activities, immunity mechanisms. Okay? So what exactly happens now? Below the CMI, write down these points. First is antigen identification, then activation of T cell, and then elimination of the, just write one word, antigen recognition, then T cell activity. Activity means proliferation. Why proliferation? Because suppose one unit of antigen has come and one T lymphocyte is present, then it can take care of. But if 10 units of antigen have come, then 1 unit of T lymphocyte will not be able to handle it. So we require proliferation. Proliferation means multiplication. Okay? And after that, elimination. See, all of you need not write in one row. You can write later on. You can take it from that person. You can copy it later on. Otherwise, we will be running short of time. Understood? Okay. These cells which are identifying these antigens are called as antigen presenting cells. Antigen presenting cells has been covered in reticular endothelial system also. There are multiple examples of APC. They are called as APC. So you are aware of these words: macrophage, dendritic cells, and B cells. You should remember this terminology APC. Just below that, below the elimination of the invader, write this APC. Later on you can read what is APC. It is called as antigen presenting cell. Is that clear? See, these are only the broad features. Later on, once you have to read. I am trying to make it as easy and as simple as possible. Okay? Now these APCs are present all over the body. They are present in liver, they are present in spleen, they are present in skin, they are present in many places. This APC can come as a short note. A short note of 5 marks, antigen presenting cell. Okay. Now see, below the APC you write these steps. What exactly is taking place? Just write the first word: ingestion, digestion, fusion, all these things. I will explain it. What is ingestion? See, the pinocytosis, phagocytosis process is basically ingestion. You know that mechanism, how it will, a capsule will be formed and it will be ingested by it will be taken inside the cell, inside the T lymphocyte. Digestion, what is the meaning of the word digestion here is breakdown of larger products into smaller products. All these antigens are proteins, big polypeptide chains. These chains are broken into smaller peptide chains and amino acids. That is the meaning of the word digestion. Then fusion, you can understand the meaning of the word fusion, there is nothing to explain. Then binding of peptide fragments, these broken fragments, smaller fragments to MHC molecule. This is something you have to understand. Write MHC, write the full form. It is called as major, MAJOR, major histocompatibility. Histo, HISTO, histocompatibility complex. Okay, this is a very important terminology in immunity. MHC, major histocompatibility complex. We will see what it is in details later on. For the time being, you remember MHC. This is the portion which will bind with the antigen. Okay, with the foreign body antigen. So that binding is called as incorporation. Incorporation, exact meaning of the word incorporate is, corporate means to get associated. Incorporate means taking inside. If somebody comes from this door into the lecture hall and sits in this class, then we will call it as incorporating that person or student. That is the meaning of incorporation. Is that clear? You should understand the meaning. Okay. See, these are diagrammatic steps. These are steps which are shown in a diagrammatic way. This is from a textbook. It is for easier understanding. Okay. All whatever we have discussed till now, this is a brief recap of the procedures. Antigen, then this is the first stage, ingestion, followed by the other stages. This is from some textbook. Most of the textbooks will be giving this type of explanation. Okay. I hope it is clear. Yes or no? Whatever I have told, it is just an explanation. If you want to read, you can read. Ingestion, you know, formation of an encapsulated structure, either pinocytosis if it is liquid or phagocytosis if it is solid, taking inside. Digestion is breaking down into smaller products. Fusion, and finally MHC complex. MHC, major histocompatibility complex, it binds with the antigen of the foreign body. So MHC can also be called as our antibody, and the protein which is there from the foreign body will be called as antigen. So antigen antibody complex. This will also come in cell mediated immunity. Okay. This is another important concept. Antigen antibody complex. Just below MHC, write down this word, antigen antibody complex. Okay. Digestion. The word digestion here always it has the same meaning, that larger products will be broken down into smaller products. Larger products, biochemical products. Polypeptides will be made into peptides. Peptides will be made into smaller chains, further into amino acids and like that. Okay, that is the meaning of the word digestion. Similarly, for carbohydrates, large chains will be made into smaller ones. Finally, glucose and fructose. Same thing for fats, cholesterol, and they will be converted into fatty acid. That is the meaning of the word digestion. See this MHC AG. This AG is antigen. MHC is antibody. How will you understand? See, with a more simpler example. We have a T lymphocyte. We will call it as T. Okay, and we have a foreign body. Some bacteria is coming and it is invading our system. It is a bacteria which is not normally present. We will call that bacteria as B. On the cell membrane of that B, there is a antigen. We will call it as B1. This B1 is totally foreign to our system. So our T will not recognize this B1. So our T will consider this as a foreign body, as an invader. It has to attack B. It will identify B by B1, by the antigen which is present on B. Is that clear? Now T will have its own system of defense. We will call it as T1. That T1 is MHC, major histocompatibility complex. So T1 and B1 will form a group. T1 will take care of B1. That is this MHC antigen complex. Understood? Can you follow this? Can you follow this? Yes or no? The foreign body, the foreign organism, bacteria or virus, will have some proteins on its cell membranes which will be infective or troublesome to our system. They will be identified as foreign antigens. They will be taken care of by our T lymphocyte MHC, T lymphocyte major histocompatibility complex. Of the initial stages, the final stage is this. It will be formed, antigen antibody complex will be formed. Okay. T cell, this T1 component and B1 component will bind at a place which is called as T cell receptor. There is a place on the T cell membrane which is called as T cell receptor. This will cause activation of, see, you have written MSC and below that you have written antigen antibody complex. Below that you write T cell receptor, because antigen antibody complex will get attached to the T cell receptor, and after the attachment at the T cell receptor, this T cell will get activated. So just below that draw an arrow and write T cell activation. So now the chain completes. The T cell is now activated and it is now competent to take care of the foreign body. Now how it will take care? The initial steps you have already seen. If the T cells are less in number, then they will multiply. So after activation, just below that you write proliferation or increase in the number of T cells. See, this is another diagrammatic representation. Try to have a look at this. Spend some time, 15 to 20 seconds, you will understand the idea of antigen, antibody, how they are associated, and the T cell receptor. Again, this is from some textbook. It is very simplified way. It will help you in understanding. Is that clear? Below the T cell, you have written T cell proliferation or multiplication. Okay. This is the event which will lead to T cell proliferation or multiplication. Okay. Should I go ahead? Again, the same thing. Net result is T cell activation and T cell proliferation. So initially the T cell was suppose 1. Now it will become 10 or 15 or 20 as the need is there. Is it clear? You know that you have done WBC count. You know how much is the T cell, how much differential leukocyte count. You know that there is lymphocyte count is about 30 to 40 out of 100. So in that field, so this lymphocyte count will now increase. A very specific increase will take place in the T lymphocyte count. The lymphocyte which is concerned with taking care of this particular bacteria B1. Accordingly, those lymphocytes will increase, not all. This is called as activation of lymphocyte. Also, you can call it as proliferation or increase in the number of lymphocytes. Selective increase in the number will take place, not all. Can you follow this? T lymphocytes have very specific duties. One lymphocyte will take care of B, other will take care of B1, other will take care of B2, something like that. Okay. Now you have written activation and proliferation of T lymphocyte. Now there will be two types of proliferations. One is cytotoxic T cell, other is memory T cell. Meaning of the word cytotoxic, cyto means cell, toxic means anything which is lethal or going to kill. So this type of T cell will directly kill that bacteria B or C or D, whatever it is. Other type is called as memory T cell. What is called the meaning of memory? Memory means to remember. This type of T cell will take an imprint of that bacteria B or C or D, and it will remember that bacteria, and it will keep that image as a memory, and whenever there is second infection by B or C or D, these memory cells will now be activated immediately. So they have prepared themselves for further attacks. So the first line of action will be by cytotoxic T cell. Later on response will be by memory T cells. See, if I ask a question to you today, you may not be able to answer it. But if I give you those set of questions and ask you to come tomorrow, then you will be coming prepared. That is the activity of memory T cell. Can you understand this? So it is taking some time to prepare against those antigens, and next time those people will come, those foreign bodies, they will be immediately taken care of by memory T cells. Understood? So below the T cell proliferation, cytotoxic T cell and memory T cell. Have you understood the concept? Cytotoxic and memory? Understood? Yes or no? Not at all difficult to understand. Memory, they will not immediately act. They will take some time. They will make a machinery which will take care of those foreign antigens. It will take some time. 1 day, 2 days, maybe more also. This is the basis for vaccination. This is the basis for vaccination. We take a vaccine, then we take a booster dose. Most of you must have been vaccinated during Corona period. Maybe you were small at that time, but at least you might be 13 years or 12 years or 14 years, something like that. You must have been given vaccination. So one vaccination was on day 1 and then vaccination was on day 30. The concept is this, memory T cell. Can you follow now? Okay. Now of the various types of T cells, which is important from a medical aspect, which you should remember always, is T8, also called as CD8 or CD4. The significance of this CD4 and CD8 is in HIV. Everybody knows what is HIV infection. In that case, that HIV virus is taken care of by this specialized type of T cell, which is called as CD4 or CD8 or T8. So whenever a person with HIV is there, monitoring of this cell count is done, CD4 and CD8. If this cell count is decreasing, it means the HIV infection is increasing. You have to remember this. This you must remember. As medical students, you have to remember this CD4 and CD8, also called as killer T cells. Okay. Just below cytotoxic, you can write killer cell or NK cell, natural killer. Okay. Below that, you write this example of CD4 and CD8. You should not forget, under no circumstance, even if you are woken up in the night at 2, you should know what is CD4, CD8 in relation to HIV. Remember this, try to revise this again and again, repeat this again and again, and it should be firmly in your memory. CD4, CD8, okay? Very, very significant. Okay, now what will happen by memory? You can read and understand. There will be multiplication of those memory cells for future activities, and immediately depends upon the type of infection, how much time will be taken, sometimes immediately, sometimes a little bit more. Multiple T cells will be formed. So when the second infection is there, second invasion is there, it is taken care of immediately. Okay, this is the significance of memory cell. How these cells act? Memory cell is, they will identify the antigen and they will produce appropriate antibodies. How cytotoxic cells will act? See, cytotoxic, they will do cytolysis. Cyto is cell, lysis is breakdown. Are you aware of this word lysis? Yes or no? You have heard this word lysis or no? Yes. Lysis means breakdown. The actual meaning of lysis is to make things liquid, from solid to liquid. That is lysis. Okay. So it will break the foreign invading cell. How it will break? It will make holes in the cell membrane of that bacteria or virus. The holes are made by enzymes which are called as perforins. Perforation means hole. Perforation means hole. So this activity is done by chemicals or enzymes which are called as perforins. So you can imagine a bottle with water, and if holes are made in that bottle, the water will come down. So that perforation is done by these chemicals called as perforins. Naturally the cell will die. So that is the mechanism of one mechanism of cytolysis. Another mechanism is osmotic lysis. You know what is osmosis? Movement of water from low gradient to higher gradient. That is also one mechanism. Once the holes are made, the water will go inside. Because the lipid bilayer, the cell membrane, that way will not allow water movement. But once holes are present, water will go inside, the cell will swell in size, it will increase in size, finally it will rupture. So the infective element is lost. Another important activity other than perforin, this you can only hear, don't try to write down. Once you develop confidence about the items you have written till now, then you can go into the details. Perforin, in addition to perforin, there is something called as lymphotoxin. It is a chemical which is released by the lymphocytes. It is a toxin for the foreign bacteria or virus. So it is called as lymphotoxin. The action is more or less similar to perforin. Okay? And of this there are multiple types. In lymphotoxins there are multiple types. If you can remember, then you should try to. Otherwise, for the timing you can omit. One or two examples you can remember, at least the groups. One of them is interferon. This is a common thing. It will come again and again in medicine and in other branches also. So you should know what is interferon. It is a type of lymphotoxin. Okay? They are secreted by multiple places. Try to understand the concept. Cytotoxic T cell. We are talking of all cytotoxic T cell activities. Perforin, osmotic lysis, lymphotoxin. In lymphotoxin, there is this factor is also there, tumor necrosis factor, and interferon. Try to understand that these are group of chemicals which are released by the lymphocytes. The specific lymphocyte which is taking care of that particular bacteria or virus. If this appears complicated, for the timing you can leave it. Try to read it at least. If you cannot follow, it is alright. Next reading you will follow. So the action of these interferons they are basically antiviral. Anything which is acting against the virus is called as antiviral. Okay. So they are of multiple types. For interferon, you can only remember that they are of multiple types and they are mainly against the viruses. Most of the antiviral activities are done by interferons. Okay. I hope you know the meaning of the word phagocytic activity. And another is opsonization. Opsonization means to attach something to that cell by which it will be identified by our immune related cell, by our T lymphocyte. Or you can say decorating it or making it more tasty so that it is identified by the T cell. So that procedure is called as opsonization. Or you can say in more medical terms, marking of that cell. Marking of that cell, the foreign body, by a product which is called as opsonin. So that procedure is called as opsonization. Okay. See if it appears, if it is difficult for you, for the timing you can leave it. A flow chart. This is a flow chart which is depicting all the activities of cell-mediated immunity after the activation of T cell. This is from some textbook. All of this is not a part of your syllabus. All of this, but you should remember that T cell activation is done. After that, there will be some chemicals which will be lymphotoxins, interferons, and likewise, and they will be having action on specific virus or bacteria. In short, this is the flow chart. Don't go into the details. It will not be helpful to you initially. Later on, if you want to go in specific details, for one particular infection, you should go in details. Later or sooner, as a storybook, you try to read it, that is HIV. In HIV, you should know some things in more details. Not all, but some things in details. Okay. Now see, below that cytotoxic cell and natural in memory cell, you write down these things. Cell-mediated immunity is taking care of these things. 1, 2, 6. Now of this, number 4 is of extreme significance. Activity in case of number 4 is against our safety, because in transplants the body is already having a system which is not functional. Suppose there is liver transplant or kidney transplant. The person who requires that transplant is not having a functional kidney or liver. So in that case, if the T cells start acting against that transplanted kidney, then that entire activity will be wasted. So we require protection against that transplanted kidney. That transplanted kidney is a foreign body, but still we want that it should be not attacked by our own T cells. In this case, we require a transplant protection. Normally what will happen is there will be a transplant rejection. That is called as transplant rejection reaction. It is because of our T cells. Because our T cells are not identifying that foreign body as our cell. So nowadays what they do is they do the MHC matching, also called as HLA, human leukocyte antigen. Human is human, leukocyte WBC antigen. So they do that HLA typing. If HLA typing is done, then there is no question of rejection. But of all these 1, 2, 3, 5 and 6 is protecting the body. But number 4 can be opposite also. We have taken liver or kidney from someone and the body is acting against that transplanted organ. So we don't want that. Okay. See this portion, number 4. We go into a different topic which is called as autoimmunity. Autoimmunity. Have you finished thyroid? Thyroid? Yes or no? Okay. Skeletal muscle? Skeletal muscle? Nerve muscle junction? Okay. There are 2 disorders which you will come across. In nerve muscle disorder, there is a disorder called as myasthenia gravis. Myasthenia gravis is an autoimmune disorder in which there are antibodies produced by our own body against our own system, against cholinergic receptors. Similarly, there is a disorder called as Graves' disease in thyroid. They are autoimmune disorders. Okay. So this transplanted cells very close to autoimmune disorders. Auto means self. The body will start reacting against our own cells. That is called autoimmune disorders. So once again from the top to bottom, immunity, types of immunity, cell mediated and the other type of immunity. Now write the name of the other type of immunity. This is the name. Only write down the name, we will see this in the next lecture. Humoral immunity. The first was cell mediated immunity. Okay. In cell mediated immunity, there is a foreign body, antigen is identified, there are steps. T cell activation will take place. 2 types of T cells, cytotoxic and natural killer. In cytotoxic, you have to remember CD4 and CD8. Then another type of cell which is called as memory cell. And finally, there are some toxins like lymphotoxin and interferons which are released by the T lymphocyte, which will take care of the foreign body. And the use of cell mediated immunity is this. I hope you have noted this. Okay. Now this is one type of immunity in which the cell is the first activity. T cell activation is the first thing to take place. In the second type of immunity, which is humoral immunity, cell activation is not the first step. Already antibodies are present. Antibody will directly attach to the antigen. So the first step is different. The rest all remains the same. In this case, the antigen was identified by the T cell and then the activity started. In humoral, there is existing antibody which is present in the body. Okay. We will see this in the next class. Here, the starting point only differs. There T cell activation took place, here antigen antibody direct binding will take place. There are 2 types of lymphocytes, T and B. T lymphocytes are mainly for cell mediated immunity and B lymphocytes are for humoral immunity. So these B lymphocytes will already secrete the antibodies. They will be forming those antibodies. These antibodies will be in blood circulation. So B lymphocytes, the humoral immunity is more immediate type of immunity. Instant type of immunity, immediate acting. Cell mediated, as you have seen, may take some time because we require proliferation, activation and memory T cells. Is that clear? This is cell mediated immunity. In short, you have to read based on the points which I have already enlisted, which most of you have noted. You read on those points. In the next lecture we will see humoral immunity. Okay? Most of the vaccines which we are taking, you must be knowing the names of oral polio vaccine, BCG vaccine, DPT vaccine, this recent corona vaccine. All these vaccines are humoral type of immunity. They give humoral type of immunity. Also, you should remember that this is also based on lymphocytes. Sometimes T lymphocytes will also contribute to the humoral immunity because the memory type of T cells will take some time and then they will produce appropriate antibodies. So, humoral immunity principally is by B lymphocytes, but role of T lymphocyte is also there. Is that clear? Is that clear? Can you follow? Yes or no?
Yes, sir.
నా ఫోన్ ఎరగట్లేదు ఎందుకు చెప్తారు నిజంగా రావురా అరే పుట్ట మేమున్నారా ఇదే